Hello, my name is Jessica Ross, and I'm working at Stanford University as a postdoc in the Keller laboratory. I also do work for the United States Department of Veterans Affairs. I use sensory motor and multisensory neuroscience for cognitive and psychiatric clinical applications, including with TMS and TMS-EEG. In our lab, one of our goals is to develop fully automated, fully personalized, and adaptive treatments for mental health disorders using TMS and EEG. We're running multiple studies on ways to optimize the TMS-EEG signal. Here are a few of our recent publications in the area, but please see my website for all publications. We are using this TMS-EEG signal to explore TMS protocol personalization for individual patient treatments. And I also explore ways in which EEG and TMS-EEG can be used to better understand and predict complex brain disorders, such as delirium, depression, and Alzheimer's disease. This talk today will focus on reducing a part of the TMS-EEG signal that is not the relevant signal, but instead the result of off-target sensory EEG confounds. For single pulse TMS-EEG, we apply a TMS pulse to the scalp and measure the resulting brain activity using an averaged EEG waveform, the TMS evoked potential, or TEP. But the EEG records more than just the direct effects of the TMS, of course. And so this stimulation brain response system has relationships within it that are much more complex than simple input-output relationships. There are contributions to the EEG that are overlapping in time, but from different sources in the brain, and may have nonlinear effects on the averaged EEG. The most obvious confounds in the TEP are from the sensory effects of TMS. What I mean is that when TMS is applied, there's a tapping sensation on the scalp, perhaps some muscle activity, perhaps some eye movements or startle, and also there's a sharp click sound produced by the snapping of the TMS coil. Brain activity from the sensory experiences during TMS can be a large part of the EEG recording that we get. This is an example of what the TEP often looks like. In this type of figure, we observe the averaged EEG following single pulses of TMS. Time zero here is the time of the TMS pulse, and the window to the right consists of a series of EEG deflections after the TMS pulse. So the figure here is an example of the commonly described TEP peaks, although we're still trying to define many of these peaks and to learn about their timing and brain sources. You may notice that the largest parts of the TEP in this example occur around 100 and 200 milliseconds following the TMS pulse. This N100 in particular has been used in a number of studies, but the understanding of exactly what it is a matter of some debate, although it is becoming understood more universally as a mixture of sensory and non-sensory cortical activity. Now I'd like to show you an EEG event-related component that occurs in the TEP, but is not related to cortical stimulation from the magnetic field under the coil. This is called the auditory evoked potential. This potential was evoked with an auditory stimulus. This was not a TMS experiment. So time zero here is the time of a short duration piano tone. For auditory neuroscientists, this is a familiar waveform and well studied to onsets of sounds. It's present in the TEP only because of the click sound that the TMS coil makes and is often the largest part of the TEP waveform. The auditory evoked potential is known to have peaks around 50 milliseconds from primary auditory cortical response and at about 100 and 200 milliseconds from responses in surrounding belt areas of A1. This potential is a response to the sound of stimulation, not to the magnetic pulse of the TMS inducing neural changes. The auditory evoked potential is also sometimes called the vertex potential, which I prefer slightly because although it is auditory, it is impacted by multisensory perception and non-modal aspects like stimulus salience. Here are some examples of what the vertex potential looks like when evoked with the TMS click sound. The time course and distribution in scalp electrodes is very stereotyped, And the amplitudes of the peaks, particularly around 100 and 200 milliseconds, can be quite large. So we're working on better understanding and modifying this vertex potential so that we can get a better look at the other responses to TMS in the EEG signal. One thing that we're studying is how the feeling of the TMS pulse tapping on the scalp interacts with the salience of the sound, and how the predictability of the timing of the sound also changes the salience of the sound, and how these things affect the size of the vertex potential. The sensory contributions to the TEP should not be ignored or interpreted as direct cortical responses to the TMS pulse. Because this waveform is often the largest part of the TEP, it is problematic because it obscures understanding of what else is happening in the EEG following the TMS pulse. In a recent review paper, we stressed the importance of evaluating the TEP for both reliability of the TEP peaks and also validity of the peaks relevant to the TMS effects. The vertex potential is a part of the TEP that is highly reliable but not valid. There are two ways for dealing with the vertex potential in TMS-EEG, and both are intended to minimize the amplitudes of the N100 and P200 peaks. This potential can either be removed or can be experimentally suppressed. I will talk about our efforts to build two tools to help with this: an ICA removal technique and a sensory neuroscience-informed suppression technique. So one approach to dealing with the vertex potential is to try to remove it from your signal, either with ICA or other techniques, or by using a sham stimulation for comparison. The vertex potential should be present with sham TMS as well as real TMS, but the intended TMS-evoked brain changes should not be present with sham TMS. So sham can be useful for identifying parts of the TEP that are likely related to the sensory aspects during a session. In one study, we optimized the use of independent components analysis for isolating the vertex potential. In this approach, we used data from a sham stimulation to improve the ability of ICA to isolate the vertex potential in data from real stimulation blocks. This method has four main parts. First, we merge the simulation blocks that all use single pulse TMS, including sham. In the two datasets we describe in this paper, we used targets at DLPFC, a parietal target, and sham. The thinking is that these should all have equivalent vertex potentials following each pulse. So this part of the signal should be shared across all these stimulation blocks. Then we use independent components analysis to separate the EEG signal into the contributing signals caused by independent sources of brain activity. ICA is a spatial filter, so it will isolate components together that have shared scalp topographies and will not be influenced by different amplitudes in the signal between conditions. Because all the conditions should have the vertex potential, this potential should emerge as one or two of the independent sources. We can identify the vertex potential using its stereotypical time course and distribution on the scalp, and then remove it from the rest of the signal. Lastly, we can unmerge the simulation blocks after removal of the vertex potential before further analysis of the TEP. In this figure, I'm showing the TEP and scalp maps before and after removal of the vertex potential using this technique. These before and after TEPs are from stimulation to DLPFC in a sample of 10 young adults. The large sensory potential, most noticeable here at approximately 100 and 200 milliseconds, is significantly smaller after removal, and the remaining brain signal appears to be complex brain signal that is distinct from the sensory potential. And here are before and after TEPs for DLPFC stimulation, parietal stimulation, and sham TMS, which should be mostly sensory. This work was done with these 10 young adults and replicated in 16 adults over the age of 65 years. In this paper, we show using two clinical data sets across different designs, evidence in cortical topography, TEP time course, global and local measures of brain activity in sensor space, source analyses, and a cosine similarity analysis that this procedure can be effective and conservative in removing the vertex potential from the TEP. We show that the TEP remaining after removal of the vertex potential is unique to stimulation site and to individual subjects, and can provide insight into TMS-evoked potentials, as well as other modulated oscillatory dynamics in the same time window of the TEP. There are a few important points I want to mention. For this to be reproducible, it should be used with clear criteria for identifying the vertex potential. These components must be shared by all the blocks, including sham, must be time-locked to the TMS pulse, must have the stereotypical midline topography that's slightly more anterior than center, and must have peak activity at approximately 50, 100, and 200 milliseconds. The biggest limitation for really any method for removing the vertex potential after data collection is that there may be nonlinear effects of sensory activity on the TEP that would not be isolated using this approach. In a recent letter to the editor of Brain Stimulation from Dr. Zeman's group, they showed a dataset in which sensory and other parts of the TEP did not appear to have nonlinear interactions. But it is likely that in many cases those interactions are present and very difficult to conclusively identify. However, I still think removal techniques can be a useful and valid approach for some studies. The question I encourage considering before using a removal technique is how might auditory processing of the click sound complicate interpretation for a study, even after removal of the vertex potential. In other work, I study EEG dynamics during auditory event processing, and we see induced oscillatory phase dynamics with predictable auditory events. So it is worth considering whether auditory event-related dynamics may have implications for your specific study. And if so, make sure to use a jittered interstimulus interval with intervals that are difficult to predict. Another approach to dealing with the vertex potential is to try to mask the sound of the TMS using an auditory noise sound. Although this can be effective if the volume of the noise is loud enough, it often is not effective, and the volume of the noise required can be uncomfortable for subjects, and particularly in patient populations getting TMS. Using foam padding between the coil and the scalp may reduce bone conduction of the sound and minimize the scalp feeling, but the optimal sensory masking technique is not known, and the vertex potential, as well as auditory perception of the TMS click sound, are usually still present after attempts to mask, particularly with the clinical prefrontal brain targets. High-intensity TMS to DLPFC is very difficult to mask. In this paper, we present a sensory suppression technique called the ATTENUATE protocol. It uses concepts from sensory neuroscience to minimize the sensory potential in the TEP during data collection. For this approach, we used a noise sound that matched the frequencies present in an auditory recording of the TMS click. However, I think other noise sounds, such as generated using the TAC tool that Matteo will present next, could be used instead. We used foam padding between the coil and the scalp. We added over-the-ear earmuffs to provide additional noise reduction. So these are 3M earmuffs designed to be used by construction workers with a noise reduction rating of 29 decibels. The model we used has a wire band that sits behind the head instead of a band that covers the top of the head, where the TMS coil might sit for mini brain targets. We also used an unjittered interstimulus interval between the TMS pulses. So most people are using jittered ISIs. The reason is usually that for single pulse TMS, we want to avoid inducing any lasting plasticity effects from unjittered repeated pulses. But by using a jittered ISI, the saliency of each TMS pulse is increased, which increases the size of the vertex potential. We went back to a more predictable ISI timing. Using ATTENUATE with 20 healthy adult subjects, we showed reduction of the vertex potential and reports of loudness and scalp feeling perception significantly more than other commonly used masking techniques. Here you can see that the vertex N100 and P200 is reduced in amplitude, and perceptual ratings of loudness and scalp feeling are also reduced. We find no difference in pain perception. We did this while keeping the earlier potentials of the TEP intact. So take a look at the early TEP. Before about 80 milliseconds, you can see that this part of the TEP has not been affected by the ATTENUATE protocol. See the paper for a closer examination of this in multiple different masking conditions. In addition, using our blocks of 80 unjittered pulses, we provide evidence that ATTENUATE did not induce plasticity. When we match on all factors except the auditory conditions, foam, or ISI timing, we find that some of these methods reduced N100, P200, or perception somewhat, but consider these reductions only minimally effective. For the full sensory suppression, we recommend using the complete ATTENUATE combination of multisensory masking and unjittered ISI. If the best option for a study is to suppress the vertex potential, we provide a better alternative that is based on sensory neuroscience for suppressing this sensory potential. In some of my other work, I study how sustained auditory noise sounds can have large impacts on motor system excitability in muscles distributed throughout the body. In this work, we use force platforms and full body motion capture to examine movement of the body during upright standing. This postural sway behavior reflects the coordinated control of muscles throughout the entire body, which is necessary to keep from falling over. We are conducting experiments with multisensory noise, predictable sounds, using dynamical systems approaches, and with TMS and EMG to better understand how distributed muscle activity in the service of a task is influenced by sounds. You may wonder what the effects of using a sustained noise sound during a TMS-EEG experiment might be on the vertex potential and also on motor excitability and other networks connected to auditory cortices. The vertex potential can be masked in certain situations, but the noise sound is likely causing unintended consequences and therefore possible confounds in brain state and motor excitability, and therefore also in the EEG. I still think we can use sensory suppression techniques such as ATTENUATE, but the question we should be asking when considering a suppression approach is whether these consequences are likely to complicate interpretation for a specific study. But for practical purposes, for TMS-EEG researchers wishing to minimize the vertex potential, should you use a removal approach like our ICA method, or should you use a suppression technique, such as the ATTENUATE protocol? To answer this question for your own lab, I encourage you to consider the following. Number one, have you already collected the data and noticed a large vertex potential in your data? Or are you planning a new study? If you've already collected the data, then a removal technique is your only option, and we provide a method using ICA that can do this effectively. If you have not collected the data yet, then you can also consider suppression techniques during data collection. And number two, which is potentially more problematic for your specific study, noise processing or click event processing? I would like to acknowledge the work of my mentors and colleagues on the studies described today and the funding sources for this work. Thank you all for your attention. Please contact me for questions or comments at jross4@stanford.edu.